Yeah, we could. Yeah, I definitely think other people can make their own groups. It doesn't have to just be one big. So yeah.
Yeah.
Yep.
Hello, how are you guys doing?
Doing well.
Doing good.
Doing
How about
good.
you?
I'm doing all right.
Good.
Let me get set up here. Are there any questions before we begin?
I do have one question, but it's related to the worksheet for lecture 2. So I don't know if you want me to ask now or at a different time.
You can ask now.
Okay, so last week when I posted on it, I posted like simultaneously with someone else. But I posted second. But then I noticed that, and then on Saturday I went back in and I answered a new question originally. Like I was the only one that answered question 8. And so I'm just wondering, because when you graded it, you said, hey, you need to do an original question, which I totally understand. But I did at the end, just not the first one that I answered.
Oh, so you're saying you answered 2 questions there?
Yeah.
All right, and I probably didn't notice that you had done 2 questions. Is that correct?
Yes, and I wasn't sure if I should delete my first one or not. I guess I should have made it clear.
Yeah, you could leave the first one. Normally I do check to see if somebody else answered another question. Um, did I mark you down on that?
Yes, that's what
Okay.
I'm asking.
So what you need to do is, oops, don't want to end the meeting here. I'm trying to get this settled here. Let's cancel that. We need to talk at another time where I can go in and correct that. Okay.
Okay, sounds good.
But I will give you credit as long as you answered a question.
Sounds good. I'll stay late after the lab tonight. We can talk about it then.
Okay, that will be a good time to do it during the lab or after I've done talking on the lab. All right.
All right, thank you so much.
Why don't I go to Clark and talk about the grading. I did get some grading done, not as much as I would have liked. Let me call that up, and I'm not showing my screen, so I can look at that and tell you what I did. But I did some grading. All right, I'm there. This is not showing correctly. So practice quiz 1B, I've still got to go in there and manually give you grades, but I gave you points for the Plagy quiz. For extra credit, study sheet. I gave most people credit. I'm not sure why there's a few people who did not get credit on that. Maybe that one wasn't, maybe you could still turn it in. I awarded most people credit. Worksheet for lecture 1 has been graded. Worksheet for lecture 2. The first grading of lab 00 and lab 01 have been graded. Lab 00, I think. Let me look on this. I thought lab 00, I may have graded the second time, but I might be mistaken on that. Lab 01 has to be graded by, meaning your corrections to lab 01. Oh, shoot. I'm not getting it now. The grading for lab 01 has to be corrected by 7:29. So you still have time to turn in your corrections for lab 1. For lab 00, if you didn't turn them in by 7:22, meaning this Saturday, I will not give you credit. So hopefully for lab 00, you'll turn in your corrections by Saturday, 7:22. If you do turn them in later, it is possible to turn it in later because I have to have one shut-off time for both of these labs. And if you do turn it in later than 7:22, you will not get credit on the second grading. I will give you a late credit, I guess, but it'll be counted as late. I think that's it for the grading. Any question on any of that?
Yeah, I didn't get the full points on the practice quiz, even though I answered the questions
Yeah, I
correctly.
haven't gone in and graded that. I need to manually go in there and assign points for that on the practice quiz. So I have not done that yet.
Oh, okay. Thank you.
Okay. All right. I know quiz 1B is due this Wednesday, I think. So you guys should take quiz 1B for 75 points. Oh, man, you have to take that starting, I think, 11:59 p.m. tonight to 11:59 p.m. Tomorrow. I'm meaning July 26th. All right, any question about any of that? Let me get out of the grading. There we go. Now I can show you my screen. All right, I didn't get any questions on that, so let's say that today is July 25th. I can blow that up a little bit. And we should be starting chapter 3. There will be a lab today, lab 4. And remember that you have to do quiz 1. Quiz 1 is going to cover chapter 1 and chapter 2. The start of chapter 2, but not the end of chapter 2. And then labs 0, lab 1, and lab 2. Any question about any of that? If not, where did we finish on chapter 2?
Sorry, I have one quick question about the quiz. So you just said that it's on lab 2. But we haven't been graded on lab 2 yet. So I mean, I'm assuming I understood it fairly well and I'm gonna do all right, but I guess I would have preferred to have your feedback prior to being quizzed on it.
Well, if you request me to start grading lab 2, I'll try and get started after the lab today. No. Oh, it looks like I finished this. Well, that's good. If you request me to start grading lab 2, I'll try and get that done after the lab today. But I probably will need a reminder, so please let me know. So we're not behind schedule. I thought we were. No, I've got that we had talked about chapter 3 that we talked about up to this slide, that we discussed that the metric system is used by all countries in the world except for 3. And oh yeah, we were supposed to start chapter 3. All right, so we are on schedule then. We're supposed to finish chapter 3 today. I don't know if we'll do that, but we will get started. All right, you should know the typical sizes of microorganisms. Their general average size of a eukaryotic cell is 10 to 500 micrometers, and it does depend on what cell you're talking about. Like the human egg is around 500 micrometers, and a chicken egg is much larger than that. A chicken yolk cell is probably the largest cell you've ever looked at. The yolk of the chicken egg is actually a cell. And there are larger cells on the planet Earth, but most people have not seen the yolk of a turkey egg. And I've never even seen the yolk of an ostrich egg. That would be the largest cell on the planet. For eukaryotic cells. For prokaryotic cells, they're smaller. The range actually goes down about 0.4 micrometers is the smallest prokaryotic cell. And then the largest is up around 10 micrometers. So the average prokaryotic cell is about 1 to 2 micrometers. Now for viruses, they're smaller than prokaryotic cell. The very largest virus is about in diameter about 0.44 micrometers in diameter. And that is about as large as the smallest prokaryotic cell. When we look at the virus length, there are some viruses that actually have a length around 1 micrometer. And that actually is Ebola. You've probably heard of that. However, Ebola is not the largest virus because it's long, but it's really skinny. Like I said, the largest virus in its diameter is actually megavirus, and it is about 0.44 micrometers in diameter. And we'll talk about the size in diameter. When we're talking about things, you should understand that there are different sizes of things. And depending on the size, you may need to use a microscope or an electron microscope to view it. Let me blow this up. So about the largest thing you need to know is a person. Obviously, an elephant's bigger than a person. And a chicken egg is the largest eukaryotic cell that you guys are familiar with. And we're talking about the yolk is the cell. Frog eggs are smaller, and that's about the smallest thing we can see with our unaided eyes. Anything smaller, we have to use a microscope. And to see most eukaryotic cells, you have to use a microscope. To see a prokaryotic cell, you have to use a microscope. To see a virus, you have to use an electron microscope because that's smaller than a prokaryotic cell. And then proteins are smaller than a virus. They're a large molecule. And then small molecules like water are smaller than a large molecule like a protein. And then atoms are smaller than a small molecule. So you should know the general relative rank of the things on this slide, but you do not need to know the absolute measurement of things on this slide. And let me state that this graph is made by our textbook author, and he never corrected this, and continues to show this graph with an error right there. That's not 1 micrometer. 1 micrometer is right there. That should be 1 millimeter right here. So to use a microscope, you should realize in the lab we use a compound microscope. That is a microscope that has 2 or more magnification lenses. It will have an ocular lens, also called the eyepiece, right here, and then an objective lens, and they're right here. They magnify the specimen And the total magnification will be the objective lens times the ocular lens. The typical microscope will have 4 compound lenses, a 4x lens, a 10x lens, 40x lens, and a 100x lens, and they magnify 4x times. The total magnification will be the objective lens times the ocular lens. In our lab, we only have 10x lens for the ocular lens. So the total magnification will be 4x times 10x or 40x times. Any question about that? You should know all of the parts of the microscope stated here and here. The ocular lens, also called the eyepiece, is here. The tube of the microscope here. This is also called the arm right there. The objective lens is here. The stage, where we put the slide here. The stage clip, you should know that, which you can see right there. Let's see if it goes there. Now, the condenser, which is right here, it puts a cone of light on the specimen right there. The iris diaphragm opens and closes how much light comes through. And for the best illumination, you want the cone of light being about equal to your field of view for your objective lens. That means with the 4x lens, you have the iris diaphragm fairly open almost all the way. So you have a large cone of light coming up. But as you then move up in power with your objective lens, you need to close down the iris diaphragm so that when you're with the 100x oil immersion lens, you have the iris diaphragm closed down so it is essentially only a pinprick of light coming through. Any question about any of that? All right, the illuminator, also called the lamp or the light source, shown here. You should know the coarse focusing knob, the big one here, the fine focusing knob, the little one here. You should know the light switch, which that is actually the rheostat. I don't see the light switch on this microscope. It might be right there. Oops. The light switch is usually turned on or off with a switch. So one side will be on, one side will be off. And then the rheostat controls how much light is coming from the lamp. And in our lab, we don't call that a rheostat. It's called the light intensity dial. And I assume in Australia, they don't use the term rheostat. And the person who did the lab is from Australia. Okay. Any questions on the parts of the microscope? Now the compound light microscope shown here, oops, let me shrink that. Shown here has 2 magnification lenses, the objective lens and the ocular lens. A simple microscope only has 1 magnification lens. This is a simple microscope that was made by Antony van Leeuwenhoek, and it only has 1 lens that magnifies. So in a sense, a simple microscope is nothing other than a fancy magnification lens. Let's see. We'll talk a little bit about that more later. The resolution of the microscope is the ability of the lens to distinguish 2 points as separate points. With a microscope, the best resolving power we can get is 0.2 micrometers, meaning 2 points have to be distinguished or be separated by 0.2 micrometers, or they cannot be distinguished as 2 points. So the best resolution we can get is that, and all points have to be separated by at least 0.2 micrometers or more, or you cannot see them as 2 distinct points. The resolution is dependent on the properties of white light. And I think this is half the wavelength of white light. Anything below half the wavelength of white light cannot distinguish it as 2 points. Let me state that also limits the best magnification you can get. The best magnification you can do is 2,000 times. That would be with the 100x oil immersion lens and a 20x ocular lens, which we don't have the best microscopes, but that would be the best microscope you can buy. And it can only magnify by 2,000 times, because any further magnification is empty magnification, because you cannot resolve anything better than 0.02 micrometers, which is on the properties of white light. So what we're talking about, we can actually distinguish these 2 points shown here as 2 separate points under a microscope, because they are the minimum distance, 200 nanometers or 0.02 micrometers apart. But these points we cannot distinguish as separate points, and they cannot be resolved because they are separated by less than 200 nanometers. Any question there? And that's what we mean by resolution. And it, in the light microscope, is limited by the properties of light. It has to do with the wavelength of white light. Any question about any of that? All right. With light, it also has a refractive index, and the refractive index is the ability of light to bend in a different media. The reason why the pencil looks bent is because at the surface here, light bends coming out of the water or going into the water. And so the pencil looks like that. This is actually important for life, because when a bird or some other creature sees a fish in the water, the fish is going to look like it's here because of the refraction of light at the surface of the water. But in reality, the fish is here. And birds know that the fish is seen here, but it's actually down there. And when they're catching the fish, they compensate for that instinctively. People have to probably learn to compensate. Because light bends, when it's coming through the air, it actually will bend on this side of the glass as it's going into the glass. And then when it comes out of the glass, light can bend. Okay, not all the rays will bend that way. Some will keep going straight. But my point is that we call the refraction of light. Whenever light moves through a medium and then changes to another medium, like from glass to air, light will refract, or it can bend. And this is showing you why we use oil. When we're looking at light coming through glass and then hitting the air, some of the light rays will not refract, and so it will continue and will be picked up by the objective lens. And some of the light will be refracted, but will be picked up by the objective lens. Some of the light will be refracted coming out of the glass, and the refraction will be so great that it will not be picked up by the objective lens. And then some of the light will be refracted and will not even come out of this side of the glass. It's refracted back into the glass. When we use oil, light is not refracted because oil has the same refractive index as glass. And so this ray will continue being this way, not refracted. And then that one will not be refracted. And then this one will not be refracted, and it will be picked up by the objective lens, where if you don't use oil, it is not picked up by the objective lens. And that one is not picked up by the objective lens. So when we use oil, we get more light rays, which are picked up by the objective lens. And that is why we get better resolution when we use oil than when we don't. And when you use the 100x lens, you should always use oil because the magnification is not better than the 40x lens. And then the 40x lens is better because you're seeing a greater depth of focus and a larger field of view than when you're doing the 100x lens. So the 100x lens, you should always use oil, or you may as well just use the 40x lens. Any question about that? And then we use oil so that light is not refracted, and then more light is picked up by the objective lens, and that improves the resolution of the scope. When we're talking about the light microscope, you should understand that we are talking about bright field illumination, which is a normal light microscope. Well, light coming through from the lamp hits the specimen and then is picked up by the objective lens. Okay, so when we see a specimen with bright field illumination, the glass or the background of the slide is very bright and it's clear or white from the light. And then the specimen will be the color of the specimen, in which case this one is dyed pink. And it's fairly bright, so we call it bright field illumination. There's also dark field illumination, which has the light giving the light rays, and then the light rays are blocked by an opaque disc. And so the glass or the background of the specimen is showing dark, and that's because there's no light coming directly from the specimen. But there are some light rays which are off at an angle, and when they hit the specimen, they are refracted into the objective lens. And so the specimen is refracting the light, and that's why the specimen is appearing, in this case, blue or purple, and the background is not refracting the light, so it is showing up dark because no light ray is actually coming from the lamp. So all of this light that we're seeing of the specimen is only light rays that would not normally enter the objective lens, but because it went through the specimen, it is refracted back into the objective lens. And that is dark field illumination. Any question about any of that? You should know the difference between dark field illumination and bright field illumination, and that bright field illumination is what we normally have in our lab with the compound light microscopes. Any question about any of that? There are some other illumination, and we're just not going to talk about them. Here we're looking at an electron microscope, and you can see it's much larger than a light microscope. It uses a beam of electrons instead of light to magnify the specimen. And the beam of electrons are focused by an electromagnetic lens, not by a glass lens. Electrons have a shorter wavelength, which gives greater resolution. And so we can see things 100,000 times smaller than with visible light, because the wavelength is 100,000 times smaller than the wavelength of light. This allows examination of very small specimens such as viruses, internal cell structures, and even going down to molecules and atoms. There are two main types of an electron microscope that you need to know, and there are more types, but there are two main types of an electron microscope. There is a transmission electron microscope shown here, the first slide, or TEM for transmission electron microscope, and then scanning electron microscope shown here. What do you notice about these two slides? Come on, somebody should be able to see something.
One's 3D and one's 2D.
Yeah, that's exactly what I was hoping somebody would notice. And that is the scanning electron microscope is in 3 dimensions. The transmission electron microscope is only in 2 dimensions. Okay. And then another difference is the transmission electron microscope allows you to focus in on the specimen, like inside the cell. You could focus at a plane on the outside of the cell, in which case it would just be, let me go here. Hopefully you guys can see that this is in 3 dimensions. You would also only be focusing on the very top. Okay. But you could focus on different planes in 2 dimensions throughout the center of the specimen. The scanning electron microscope only sees the surface of an object. And here we're only seeing the surface of this cell. And here we're looking at the inside of this cell. So that explains how transmission electron microscopes differ mainly from scanning electron microscopes. Any question about any of that? All right. Oops, wrong one. Here we're looking at the scanning electron microscope, seeing it in 3D. And this is the transmission electron microscope. I believe that's the membrane of the chloroplast of a cell. Not that you need to know that, but just to inform you what it is. All right, a third type of electron microscope, which you do not need to know and you will not be examined on it, is scanning tunneling microscope. It uses a metal probe to scan a specimen, and the resolution can get down to 1/100th the size of an atom. So very small. And then there's atomic force microscope. The fourth type of electron microscope uses a metal and diamond probe inserted into the specimen, produces a 3D image of the specimen. When we're looking at a specimen with a light microscope, I'm going to ask you which of the cells are easier to view, the one on the left or the one on the right? Come on.
The one on the right.
The one on the right is easier to view. The only difference between these two slides is the cells here have been stained. These are unstained. All staining does is increase the contrast between the specimen and its background, that's the glass of the slide, making the specimen easier to view. And that is all staining does, increasing the contrast of the cell against its background. Any question about any of that? So with a bright field compound microscope, most microbes are colorless and have little contrast with the slide or the medium they are in. To see them well, you need to stain them. Staining adds a coloring of the specimen or the background to increase the visibility or the contrast between the specimen and the background of the slide. A smear, it is a thin film of solution of microbes put on a slide. And to make a bacterial smear, you put a thin solution of bacteria, smear them all over the slide, and we call that a bacterial smear. There is no staining or no steps involved in making a smear. And I always get students answering this question wrong, so I'm going to emphasize it again. And that is, when we make the smear, all you're doing is putting the specimen microbes on the slide. Okay? You do not do any staining when you're making the smear. So a bacterial smear is usually done prior to staining the slide. Microbes on the smear are usually fixed to the slide. So usually what we do is we smear them on the slide, let it air dry, and then we heat fix the microbes to the slide so they do not wash off at a later step, such as when you're applying the solution of the dye. Is usually in a liquid like water, and that can lift off the cells from the slide if you do not air dry them and heat fix them to the slide. And then the water washing later would especially wash off the slides. Any question about any of that? All right. So the stain, also called the dye, is adding a salt, which are two ions. So a molecule that separates into two ions, and the salt positive or negative ion is colored. Only one is usually colored. If the chromophore, the colored part of the molecule, is on the positive charge, or the cation, we call that a basic dye. If, on the other hand, the chromophore is on the anion, the negative part of the molecule, then it is an acidic dye. Any question about any of that? So we have two types of dyes, acidic dyes and basic dyes. Bacteria have a slightly negative charge. Therefore, a basic dye is a little better for staining the cell because the positive cation of the basic dye will attach to the cell and then color the slide. Most of the dyes we have in microbiology are basic dyes like crystal violet, methylene blue, malachite green, and safranin, just to name a few. An acidic dye typically stains the background of the slide. It does not stain the microbes. And that's because bacteria have a negative charge. The acidic dye will be repulsed from the bacteria, but it will stain the background, the glass of the slide, not the cells. Because the background will stain dark instead of the cell, we call using acidic dye, we call that a negative stain, and that comes from photography when we had what do they call that type of, a negative from the photograph. So the photograph would show you the colors in the correct color. If it's black and white, it would show you the correct color, meaning light in places where the person's face was light and dark where the person's face was light. So the whites of the eyes will appear white, and then the pupil will be darker. The hair would be darker. The staining the background will color the cell in the opposite, and that would be the negative of a photograph, which was showing you the image in negative colors. So where the person's face looks white, like the whites of the eye, it will actually on the negative be dark. Any question about that? And that's what we call that negative staining. And I do realize that in today's modern photography, we no longer have a negative. What was that called? When you got the pictures and they give you the negative, that was a Polaroid film, maybe something like that. All right, so this is showing you the cell stained with a basic dye. This is showing you the cell stained with a negative dye where it stains the background. You can still see the cells though with a negative stain. Any question about any of that? All right. There are three main types of stains. We'll talk about each of them. There's a simple stain, there are differential stains, and there are special stains. A simple stain is where we're applying a single dye in an aqueous or alcohol solution. The primary purpose is just to increase the contrast between the specimen and the background of the slide. The stain is left on the slide for a certain length of time, and then it's usually washed off, dried, and then examined. Now here we're looking at a simple stain, coloring the cells. A mordant may be used to intensify the stain. The functions of a mordant include: it increases the affinity of the stain in the specimen. It may coat the specimen many times to enhance it, therefore making it easier to see something. It is not a stain itself, a mordant. Here we're using a mordant which coats the specimen many, many times, and that way then we can see the flagella. Because if we just stained it with a stain, the flagella will not be visible. But this coats the specimen many, many times, and then the flagella appear. A mordant we'll talk about in the gram stain simply intensifies the staining of the first stain of the gram stain. There are also differential stains, and with these, they stain the specimen differently depending on what cell we are looking at. So two different cells, two different types of cells may stain differently. A differential stain usually uses two different dyes, but that is not absolutely required. You just need to stain the cells differently. The most commonly used differential stains are the gram stain and the acid-fast stain. And we'll talk briefly about the acid-fast stain. We'll talk about the gram stain in great detail. The differential stain known as the gram stain was developed in 1884. You don't need to know the date, but you do need to know it was invented by Hans Christian Gram, a doctor who was trying to make it easier to look at a bacterial infection in a patient. And he developed this stain so that the bacteria stained dark, and then the patient's own cells stained light, or the bacteria stained purplish, and then the patient's own cells stained pinkish. And so he did intend it as a differential stain, but microbiologists started using the Gram stain, and they very quickly realized that the Gram stain stained bacteria into two different types: the Gram-positive cells, which stain dark purple, and the Gram-negative cells, which stain pinkish. And so we use this as a differential stain, a little different than Hans Christian Gram intended, but we do use it as a differential stain to separate bacteria into two very large groups: Gram-positive and Gram-negative cells. To do the Gram stain in brief, you make a bacterial smear on the glass slide. And this is not part of the staining procedure. That is making the bacterial smear. Then you heat fix the cells onto the slide. Actually, you should also air dry it before you do the heat fixing, and then heat fix the cells onto the slide. That also is not part of the stain. It's just something we have to do on the Gram stain because the staining procedure will wash off the cells from the slide if you don't air dry and heat fix. You then flood the slide with crystal violet for about a minute. You then wash off the crystal violet with cold water. You then flood with iodine. Iodine is a mordant. It intensifies the stain of crystal violet. We'll talk a little bit more about why in a few minutes, actually. We then wash off the iodine with water, flood the cell with acetone ethanol. This is known as the decolorizing agent. You apply the decolorizing agent for only about 5 seconds. We'll talk about why in a little bit. You then wash immediately with water. Notice how I didn't say you need to wash the crystal violet off immediately with water, because you don't need to wash it off immediately with water. But here, the decolorizing agent will continue to work, and so we need to wash it off immediately. You then flood the slide with safranin. Safranin is the counter stain because it's the second stain applied, and it's a different color than the first stain. You apply that one for 1 to 2 minutes. Then wash that off, blot dry, and examine the slide under the microscope. Any questions about the Gram stain procedure in brief? All right, this is a very important slide showing you the different color of the cells at different steps of the Gram stain. Initially, when we apply crystal violet, the cells will be bluish in color, bluish purple. We then apply iodine, the mordant, and the cells will be a little darker purple and a little more purple, meaning a little less blue purple, a little more purple in color. We then apply the decolorizing agent, and that pulls out the crystal violet iodine complex from the Gram-negative cells. But if it's done correctly, it does not pull it out of the Gram-positive cells. So the Gram-positive cells will stain purple, and then the Gram-negative cells will be clear at this stage. We then apply safranin, and that's a reddish or pinkish stain, and it will color the Gram-negative cells pinkish. Any question about any of that?
Yes, I have a
Yeah.
question. Is there a reason why the iodine doesn't actually cause like any issues with the crystal violet or the saffron and create any color issues there, I guess. I don't know best.
It does
access.
change the color
Okay.
a little bit. Like I said, here it's going to be a blue purple color. This will be more of a purple color. Iodine is not really a dye, it is a mordant. And what
Okay.
it is doing is it's binding to the crystal violet, making a bigger molecule,
Got it. Okay.
and
That
that
makes sense.
intensifies the color because it's a bigger molecule in the cell.
Okay. Thank
And
you.
then it makes it less likely that the crystal violet iodine complex will wash out of the gram-positive cells because it's a little bigger. But it does wash out of the gram-negative cells. Okay, any other questions? I thought you asked something about the safranin too.
No, that was it really.
Okay.
Kind of more clear
right.
to me. Thank you.
I'll ask you a question about the safranin in just a little bit. All right, let's go to the gram stain in greater detail here. So first you make the bacterial smear on the slide, you air dry and heat fix it. And then you flood with crystal violet for 1 minute. I always say 1 minute because for all of my staining, I simply use 1 minute. After 1 minute, you then dump the crystal violet off and then wash the crystal violet off with cold water. Crystal violet sticks to the peptidoglycan in the bacterial cell wall. It's considered the primary stain since it colors all cells the same. So both the gram-negative and the gram-positive cells will stain purple or blue-purple exactly. You don't need to know that, but purple. So both the gram-positive cells and the gram-negative cells are going to be staining purple after applying the crystal violet. And we can say it blue-purple if you want. We then dump the crystal violet off, rinse the slide with cold water, and then add the iodine. Iodine is a mordant. It complexes with the crystal violet, increasing the size of the complex and increasing the intensity of the dye a little bit. The color, well, I guess what we do is we then leave the iodine on for another minute and then dump it off and then wash the slide gently with cold water. After the application of the iodine, the color is just a more purple color for gram-positive cells and a purple color or purple color for gram-negative cells. The point is the gram-negative and the gram-positive have the same color. Okay. And it's not terribly important. It's slightly different color than the crystal violet alone. All right, after that, you dump the iodine off, rinse it with water, and then after the water is off the slide, you apply the decolorizing agent. The decolorizing agent can change, but usually we use acetone ethanol mixture. You could use an acid ethanol mixture. And if you use these decolorizing agents, you only apply it for about 5 seconds. It is possible, you don't need to know this, that you can just use ethanol for your decolorizing agent, in which case you have to apply the ethanol for a longer period of time. And I don't remember how long you apply ethanol. It's something like 15 seconds or maybe even a minute. So you're using ethanol longer than you would acetone ethanol. I'm going to just talk about acetone ethanol because in our lab, that's what we mainly use for a decolorizing agent. The decolorizing agent washes the crystal violet iodine complex out of the gram-negative cells, but not out of the gram-positive cells, as long as it is done correctly. It is considered the differential step in the gram stain because after this step, the cells appear different. So after this step, the gram-positive cells will be a purple color. And after this step, the gram-negative cells will be colorless. After applying the decolorizing agent, you then immediately wash with water to wash the decolorizing agent off of the cells. Because even if you dump the decolorizing agent off, there will be trace amounts of decolorizing agent on the cells, and the decolorizing agent will continue to work. And that can cause problems. We'll talk about that in a little bit. I don't want to talk about it now because I'm just going through the gram procedure. We then apply safranin. Safranin is a pink dye. It does stain all bacteria. It is a counter stain because it has a different color to the primary stain, meaning it has a different color than crystal violet. Safranin is a pink color. You flood the slide with safranin for a minute. As long as the safranin is intense enough, a minute is long enough. If the safranin is a little weak, you can leave it on for 2 minutes so it'll stain the cells a little better. All the safranin I've always used at Clark College, a minute was sufficient time to apply the safranin. So the gram-positive cells stain purple, the gram-negative cells will stain pink after applying the safranin. You then dump the safranin off and then wash the cells gently with cold water, and then blot the cells in bibulus paper, which is a very thick paper. I'm trying to get an example here. This is obviously not bibulus paper, but it's going to illustrate the point. And the best thing to do is to get your slide and then fold the bibulus paper and then just blot it down. Do not rub the slide against the bibulus paper or rub the bibulus paper against the slide, but you want to blot it down. I don't know if you can see this or not. Just blot it down. And I like to fold it and then just blot it down so you're getting the water on both sides of the slide. Any questions about that? Well, here's a question for you since nobody got it. I state right here that the safranin gets in all the bacteria, but only the gram-negative cells appear pink. Why do the gram-positive cells not appear pink if the safranin is getting in them?
Their cell wall is different.
Their
Isn't it-
cell wall
does-
is different, but why is
Doesn't
why
it,
is
is it
the
because it has more
gram-positive?
sugar?
Hmm?
I don't know. I'm making things up, but I said, isn't it because they have more sugar in the cell
Well,
wall? But
it is true that they have differences in their cell wall. We'll talk about that. But my point is that the safranin is getting in the gram-positive cells. Why isn't it appearing in the gram-positive cells?
The purple's stronger than the pink.
Ah, that's right. You've taken microbiology before or what?
Just read ahead a little bit.
You read ahead of it a little bit. Okay. Yeah, the purple does get in the gram-positive cells, but to our eye, we only see the purple color. To see the pink color, you'd have to put the cells under the electron microscope. And if you did, let's pretend this is the entire cell here. This part would stain purple. Maybe this part would stain purple. This part would stain pink. So under the electron microscope, you can see pink in the gram-positive cells, but the purple is a more intense color. And so to our eyes, it only shows purple. And that's just the way human eyes are. We don't see the pink because it's, I don't know, overshadowed or something by the purple. But it is there, okay? We just can't see it unless we use the electron microscope. All right, I already talked about that. Oh, once you blot the slide dry, it may not remove all the water, but you do remove enough of the water that you can observe the slide under the microscope and even use oil on the slide. Enough of the water is removed, you can even use oil. Okay, gram-positive cells are purple, the gram-negative cells are pink after the procedure. Why? What's the difference in the differential staining? It has to do with differences in the cell wall. The gram-positive cells have a thick peptidoglycan layer with disaccharides and amino acids. We'll talk a little bit more about peptidoglycan in a later lesson. And the thick peptidoglycan layer does not allow the crystal violet iodine complex to wash out of the thick peptidoglycan layer. It gets trapped in there. As long as you are doing the wash quickly or briefly, meaning about 5 seconds. And I forgot to show you how you do the wash. We'll talk about that in a little bit. The gram-negative cells do have peptidoglycan in their cell wall, but it's a thinner layer of peptidoglycan. And the crystal violet iodine complex can wash out of this thinner layer of peptidoglycan. When we're looking at the cell walls, the gram-positive cells look like this. Let me blow this up. So we're looking at the cell wall there. It has a thick purple layer, which is the cell wall. The yellow is the cell membrane right there, and it's a lipid bilayer. The cell wall is very thick. It's only showing you 3 layers of peptidoglycan. It's actually over 100 layers of peptidoglycan. It may be up to 1,000 layers, depending on the cell wall and the species. Sorry, I'm getting a frog in my throat. So only 3 are shown. That's not correct. It's, like I said, actually around 100 to over 1,000 layers of peptidoglycan. In the gram-negative cell wall, it has a lipid layer, which is the cell membrane shown here and here, a lipid bilayer. And then it does have peptidoglycan, but it's a much thinner layer of peptidoglycan. And then it has a lipid layer outside the peptidoglycan. And that lipid layer we call the outer membrane of the cell wall. And it actually is a lipid bilayer, the same as similar to the cell membrane. And there's the peptidoglycan layer. Here it's only showing you 1 layer. It's always more than 1 layer. It's something like 3 layers of peptidoglycan to something like 20 layers of peptidoglycan. Okay, but it's only showing you 1 layer. Any question about any of that? And it's the thickness of the peptidoglycan layer that makes the differential and why the crystal violet iodine is not washing out of the gram-positive cells, but is washing out of the gram-negative cells. All right, I've already talked about that. I've already talked about that. So here we're looking at the gram stain of a mixture of cells, gram-positive rods, gram-negative. There's a gram-negative rod, some gram-positive cocci right there, gram-negative spirilla right here, gram-negative vibrio right there. When we do the gram stain, we sometimes get complications. Like, what do we do here? Is this gram positive or gram negative? Well, what you do is these are bad regions of the slide, and those actually are not cells. What that is a crystal of crystal violet. And crystal violet sometimes crystallizes out of solution. And then in this bottle of crystal violet, there will be a crystal, and you actually add it to the slide. And usually you can kind of see it right here. The cells actually retain, and right there too, the purple of the crystal violet. And then further out, they're pinkish. And so you don't read it in the bad regions of the slide, which are circled here and here. There's one over there too. You read it in a good region of the slide. And a really good region of the slide is this region here too. But here it's showing you a good region. Oops. Sorry, I gotta shrink this a little. Ah, crud. Good region of the slide. So this is a gram-negative cells. The gram stain is not as clear-cut as theory would suggest. Sometimes you'll be pulling your hair out because the gram stain isn't working well. To do a good gram stain, it must be performed on young, freshly growing cells. If the cells are older than 2 days old, the gram stain may not work correctly. Gram stain is also not universally applicable. Some stains stain poorly. I would say they do stain with the gram stain, but they don't take up the stain very well. So there are some problems with the gram stain, which may become apparent to you when you do your unknown project later this term. The structure of the gram-positive and gram-negative cell walls does affect the cells in other ways besides staining. It also affects how the cell responds to certain antibiotics. Like penicillin G can easily kill the gram-positive bacteria, but it does not kill the gram-negative bacteria. The reason why is the penicillin G cannot penetrate the lipopolysaccharide layer of the outer membrane of the cell wall. Let me show you that here. Let's blow this up. Oh, that one. So it cannot get through this outer membrane of the cell wall, and penicillin G actually does damage to the peptidoglycan. And if it can't get to the peptidoglycan, penicillin G isn't going to work. And penicillin G cannot work on gram-negative cells, but it does work on gram-positive cells. Any question about that? There are some other antibiotics that work similar to penicillin G, like penicillin V or cephalosporin, and some detergents. They just don't get through the outer membrane of the gram-negative cell. Gram-negative bacteria are more resistant to these antimicrobials. Okay, that doesn't mean they're more anti-resistant to any antimicrobial like not penicillin G, sorry. What's that antibiotic called? No, I'm drawing a blank here. There is an antibiotic which only works on gram-negative cells. And we can use that antibiotic to treat a gram-negative infection. You would not use penicillin G because penicillin G would not work on a gram-negative cell. And I forgot the name of that antibiotic. Sorry, I'll try and remember that. And hopefully I'll be able to look it up and, oh, polymyxin B. There it is. There's the antibiotic. It works only on gram-negative cells. Doesn't work on gram-positive cells. So there are antibiotics which work only on gram-negative cells. And then there are some like penicillin G that only work on gram-positive cells. And then there's some like ampicillin, the third member of the penicillin family to be discovered, that actually works on both gram-negative and gram-positive cells. Any question about any of that? All right, let's briefly talk about special stains. I'm not going to examine you on that, but you do need to know about special stains because you may need to do the special stain on your unknown project. A special stain, one that we're going to talk about, is a capsule stain, a stain used to illuminate the capsule. And it usually doesn't stain the capsule, it usually stains the cells and the background of the slide, but it does not stain the capsule. And like I said, some of you may need to do this special stain, the capsule stain, on your unknown project. Here we're looking at a special stain, and it's staining both the background of the slide, a light purple, and then the cell, a dark purple, and then the capsule is unstained. So we're seeing the capsule around this cell here. And I don't know if those are Coccobacillus or cocci. That looks like a bacillus, so it's probably a Coccobacillus. Here's a negative stain. It's actually India ink. It stains the background of the cell very dark color, and then it lightly stains the cells a light color. Once again, the capsule is unstained, so it's very easy to see the capsule. Any question about any of this? All right, I've actually got time to talk about one other thing, and that is I forgot to mention when you're actually doing adding the decolorizing agent. Where do I have that here? When you're adding the decolorizing agent, the best thing to do is not to time it for adding it by 5 seconds. You add the agent across the slide, holding the slide at an angle so the decolorizing agent is running down. And you add the decolorizing agent until the purple of the crystal violet iodine complex stops streaking. Once it stops streaking, you stop adding the decolorizing agent. Okay? Because if you apply the decolorizing agent for too long, it will remove the crystal violet iodine from a gram-positive cell. But if you don't apply it for long enough, you will not remove the crystal violet iodine from the gram-negative cells. So applying the decolorizing agent is very tricky, and it takes a little bit of practice. Any question about that? All right, I think we finished this lesson. And actually, I think the class continues because this is the summer term. It's a longer class. Yeah, we go to 6:50. I used to break at 6:15, so I was getting ready to stop, but we're going to continue because I think I'm supposed to start the next lesson, next chapter. Yeah, let's begin chapter 5. I'm going to take a 5-minute break. So let's go ahead and take a 5-minute break here. And I'm not going to stop the Zoom, but I'm going to stop recording here. Let me share my screen. So we're going to begin chapter 4. Any questions? Let's close down chapter 3. All right, here we're seeing a introductory slide on chapter 4, the functional anatomy of prokaryotic and eukaryotic cells. You have had the anatomy of eukaryotic cells in your preliminary course. So if I run out of time, I'm not going to cover that. But if we do have time, I will cover it. And usually I do not have time. Any questions about any of that? So the eukaryotic part should be a review. I will cover the functional anatomy of prokaryotic cells, and I will cover the endosymbiotic theory, which is actually talking about eukaryotic cells. Any questions? Sorry, I got something on my finger here. I wipe my hands. All right. In this slide, I have the major goals and a rough outline. Learn the terms. Okay, so obviously do that. And then 2, understand how a gram-positive and gram-negative cell walls differ. We'll go into that again and in a little more detail. And then 3, be able to describe the importance of taxis in bacteria. Any questions about what we're covering? Let me make sure I'm recording. Yeah, I am. All right. All right, this slide is showing you some differences between prokaryotes and eukaryotes. A prokaryote is Greek for actually pre-nucleus, and literally it means pre-kernel. Kernel, and the kernel being the nucleus. And then eukaryote is Greek for true nucleus, or true kernel, literally. Prokaryotes generally have one chromosome, and it's a circular chromosome right here, where the chromosome ends at the same place the chromosome begins. So it's a circle. The chromosomes are not in a nucleus. For eukaryotes, the chromosomes are in a nucleus, and there are usually multiple chromosomes, and they are linear chromosomes. So like for the X chromosome, which I'm drawing here, we have a beginning spot here on the X, and then an ending spot here. It is not in a circle. Any questions about any of that? Prokaryotes do not have histone, which is a protein, associated with their DNA. Eukaryotes do. In fact, the DNA of eukaryotes is wrapped around the histone protein. And that is the main protein in the DNA of eukaryotic cells, the histone protein. And like I said, it's actually, I don't know, we're going to pretend this is a chromosome here and this is the histone protein. The DNA actually wraps around it twice and then moves on to the next histone protein. The DNA of prokaryotes do not have histone proteins. They do have protein in their chromosome. And so both a prokaryote chromosome and a eukaryote chromosome is made of 2 molecules, DNA and protein. But in eukaryotes, the main protein is the histone protein. Any question about any of that? Okay, prokaryotes do not have any organelles, and by organelles, I mean membrane-bound organelles. Eukaryotes do. They do have membrane-bound organelles, and these membrane-bound organelles divide up the cell into functional units, and usually each a membrane-bound organelle performs a specific function for the cell. peptidoglycan is found in the cell walls of bacteria and only bacteria. It is not found in the cell walls of archaea. And there is no peptidoglycan in the cell walls of eukaryote cells. And of course, animal cells don't even have a cell wall. So we don't have any peptidoglycan either. Prokaryotes divide by binary fission. That means the cell literally will divide into two. So one cell will divide into two by binary fission. Eukaryotes, their cells divide by mitosis. And we're not going to talk about mitosis. Hopefully you have that in your preliminary biology class. All right, first we're going to talk about prokaryotes, the cell shape and their arrangement, their cellular growth arrangement. We'll talk about cell structures. We'll talk about movement of materials across the membranes. In eukaryotes, we'll talk about cell structures, and we really won't talk about movement of material across the membrane. And then we'll talk about the endosymbiotic theory. So we will cover that. The prokaryotes have a specific size and shape. The average size is really 1 to 2 micrometers, but they can be as small as 0.4 micrometers and about as large as 10 micrometers. The prokaryotic cells have 3 basic shapes. They can be round, and we call those coccus. They can be a rod shape, and those we call bacillus, although in truth, we also call those rods. So bacillus and rod both are scientific terms. And then there are spirals shown here. These are the 3 basic shapes of prokaryotes. There are other shapes, but generally they are some combination of these shapes. Okay, so like the coccobacillus, I'm going to show you that now because it's in the wrong slide. Right there and right there. Let me blow this up. That is a shape. Sorry, I'm not seeing where to go. Here we go. This is a shape also shown here. And you notice that, is that a cocci? Is that a bacillus? It's in between a coccus and a bacillus. And these we call coccobacillus. A vibrio is a spiral, or you could call it a curved rod, either one. And this slide is showing you that there are different types of spirals. There is a spirillum, the normal spiral, and it's a little more rigid. It does have a corkscrew movement, like it moves like a corkscrew, if you know how a corkscrew moves. I'll show you that in a little bit. And then a spirochete, which also has the corkscrew movement. It simply has more ridges or bending. It is a little more flexible, the spirochete, from the spiral. But it's just more spirals in the spirochete. There are some prokaryotes with different shapes, which are unusual, like the star-shaped Stella bacteria shown here. Actually does look like a star. That's the artist picture. This is the actual electron microscopic image. And then the Haloarcula, which is an archaea, which is actually they say square shape, but it's actually a rectangle. And there's one there. Most bacteria are not this unusual shapes. Okay, most bacteria species, all the members of the species are more monomorphic, meaning they all have the same shape, like the Haloarcula, all of them are like rectangles. Micrococcus luteus, all of them are coccus. But there are some bacteria species where the species themselves are pleomorphic, where they take on many different shapes. Most of the bacteria, which are pleomorphic, are bacteria that lack a cell wall. And there is one family of bacteria which lack a cell wall. And we'll talk a little bit about this family later in the term. The bacteria of this species takes on many different shapes. And here is a picture of some of the different shapes they have. And they don't have a cell wall, so they can take on many different shapes. The cell wall gives the shape of the cell. Okay, so that's the shape of cells. Now let's talk about the cell arrangement, also called the cellular growth arrangement. That is how the cells stick to each other. And there's different cell arrangements that the cells can take. This is showing you the cocci, which have the most cellular growth arrangements or the most cell arrangements. First, it's not stated here, but it's shown here, and that is the cocci. Cocci is plural for coccus. Coccus is singular for cocci. You can have singlets, single cocci on your slide. And when they divide, they can stay as singlets, meaning they divide and separate, or they can stay together. Two cocci together we call diplococci. Many cocci together we call streptococci. And you need 3 or more to become a streptococci. 4 tetrads together we call a tetrad, meaning 4 cells stuck together. And then 8 cells stuck together we call a sarcenia. And let's see if we can load this up. This is actually diplococci. You can see 2 cells right there. There's 2 cells right there. There's 2 cells right there. Streptococci right here. And then that's another one. This one's hard to see, but that's 4 there. And there's 4 there. Sarcenia, it's really hard to see. There's 8. It's very hard to see all 8 of them. It would help if they didn't show you a picture with so many cells, because this is another sarcenia there and another one there and another one there. If you only saw one, it'd be easier to see. And then there's staphylococci, where the cells literally look like a cluster of grapes. And actually, that's what staphylococci means. It means cocci, meaning round cells that grow like a cluster of grapes. Okay. And the cells just grow every which way. Sarcenia, the cells divide on 3 different planes. Come on, mouse. The X plane, the Y plane, and then the Z plane. The tetrads only divide on the X plane and the Y plane. The streptococci and the diplococci only divide on one plane. The singlets can divide on any plane. It's just the cells don't stick together. Any question about any of that? All right. The bacilli have fewer cellular growth arrangements than the cocci. You can have singlet bacillus, where the cells grow and reproduce, but then they become singlets. There can be diplobacilli, and that's 2 bacilli stuck together. And and they form lengthwise, meaning they're stuck on their short end. And then there's streptobacilli, which formed like sausage links. If you've ever seen hot dogs or sausage stuck together like this, where they're once again stuck together on their, my mouse has died, stuck together on their short link. Streptobacilli. This is showing you streptobacilli there. That looks like diplobacilli right there. Coccobacillus is not a cellular growth arrangement, so this should not even be in this slide. I don't know why the author put this in this slide. Okay. And then there is a bacilli stuck together on their long side. And this we call palisade, because if you flip it that way, it looks like a palisade fence. Let me end the show here and bring out paint. And if you ever read Mark Twain, he was, it was Mark Twain who was painting a palisade fence, and he was trying to tell Huckleberry Finn how fun it was to get him to paint it for him. But if you have the fence where the boards are stuck in the ground this way, this was the palisade fence, and that's the way the palisade bacteria grow. I'm not drawing that too straight, but you're getting the idea. This we call a palisade. Any questions about any of that? Where the bacilli are stuck together on their long end. And those are the only cell growth arrangement that bacilli have. The singlets, the diplobacilli, the streptobacilli, and the palisade. Any questions about any of that? Now, which growth arrangement do the spirochetes get? Spirochetes in the spirillum only have singlets. If you see a cellular growth arrangement in your cell, you will tend to see more than one. For example, with the streptobacilli can break, in which case we have diplobacilli. And then the diplobacilli can break, and then we have singlets. So for the streptococci, did I say streptobacilli? I mean streptococci. We can find streptococci, diplococci, and singlets. What you do is you give the highest order that you see. So that would be streptococci. Staphylococci can have sarxinia, can have tetrads. They don't usually have streptococci, although it's possible. And then they can have diplococci as well as singlets. And if you see a clump, you say it's staphylococci. Now, with that stated, that the growth arrangement is usually more than one, the only time you'll only see one is if the cells only have singlets. Singlets can only be singlets. They will never form diplococci and then streptococci, etc. Okay? And usually the spirals and the spirochetes will be only singlets. And so that's all you see. Any question about any of that? Now, with that stated, let me state, you need to know this for the unknown project. You don't need to know it before that. But if you want to talk about the cellular growth arrangement, you have to have the cells growing in a liquid culture and then observe the cells with a simple stain, because the staining process itself can break up the cells. And if you grab the cells from a solid culture, like growing on an agar plate, the cells will tend to stick together. And so you will not get the true growth arrangement. So when you're getting the gram cells, you'll get the cells from a solid agar plate, in which case you cannot determine the cellular growth arrangement. You have to grow the cells in a liquid culture and then stain the cells gently to look at the cellular growth arrangement. Any question about any of that? All right. Let's talk about some of the structures of a typical prokaryotic cell. All of the structures shown in red are parts of prokaryotes that are in all prokaryotes. The cytoplasm is actually in all cells, the ribosomes in all cells, although the ribosomes differ between a prokaryote and a eukaryote ribosome. The plasma membrane is in all cells, both prokaryotes and eukaryotes. And then DNA is a region or is a molecule found in all cells. Okay. The other structures may not be found in all prokaryote cells. All right, any question about any of this? I think we're close to ending here. Maybe we have 5 more minutes. Yeah, we got 5 more minutes. So let me talk about one more slide. The nucleoid region of a prokaryotic cell is simply where the DNA is floating in the cytoplasm. It does stain a little differently, which is why it's called the nucleoid region, but the DNA is not separated from the cytoplasm by anything. It's wherever the DNA is floating is where we call the nucleoid region. And obviously, the DNA can move around in the cell. And so, I don't know, there's no distinction between the cytoplasm and the DNA of a prokaryotic cell, but the region does stain different, and we call that the nucleoid region. The glycocalyx is something that is secreted by many cells, including many prokaryotes, but not all cells. It is something outside the cell wall, so we call it something outside the cell. The last part of the cell is the cell wall. Anything exterior to the cell wall is external to the cell. The glycocalyx is usually sticky, and there are 3 different types of glycocalyxes. There's the capsule, which is a neatly organized structure. It can prevent phagocytosis. If you can see it under the light microscope, then it is a capsule. The slime layer is unorganized, loose structure. It's mostly water. So that's how it differs from the capsule. It's mostly water. The capsule does have other molecules in it. The capsule also is mostly water, but it does have significant amounts of other molecules in it. And the slime layer has only a few molecules and water in it. The slime layer you cannot see with the light microscope. To see the slime layer, you have to use the electron microscope. And then there's a third glycocalyx, and it's found in biofilms. It's the extracellular polysaccharide, which is secreted by cells that form the biofilm. Actually, the extracellular polysaccharide is secreted by each of the cells in the biofilm, and the extracellular polysaccharide creates the biofilm that has cells inside of it. We'll talk about a little bit about biofilms later in the term. Any questions about glycocalyxes? And you can pronounce that glycocalyx or calyx. All right, any questions? If not, I'm going to end it here and I'll see you at 7 for the lab. All right, bye.